Welcome to part 2 of my presentation on autoimmune disease and its connection to the microbiome. In the first video, we touched on a number of different factors, but spent most of the time on molecular mimicry, immune dysregulation, and genetic propensity. As I think you're starting to appreciate, not only is the microbiome complex, but the immune system, at least to me, is mind-boggling. There are so many things we could discuss. Each topic is a presentation in itself. Aspects of our biochemistry that you would never think would exist. Obviously, we can't get into the minutiae. And we even have to leave out of this video some things that those with knowledge may like to see, such as citrullination and subsequent anti-citrullinated protein antibodies or bacterial amyloid-DNA complexes. Also, your specific autoimmune disease, if you have one, may not be highlighted in this presentation. Again, we can't cover everything. I touch on the main points and the most prevalent diseases, combined with whatever data is or is not available. I have picked out a number of papers which either highlight an interesting new topic or go on to reinforce points which I think are important for you to understand. This way the video is applicable and valuable for the most amount of people possible. So let's begin. We start with my meta-analysis for all of the studies which I have analyzed over the many years as the Director of Medical Education for a microbiome firm and now my own. If you're not familiar with this chart of mine, let me explain. This is the microbial fingerprint of autoimmune disease taken from original research in humans analyzing the fecal microbiome of those with autoimmune disease versus healthy controls. At the bottom, the x-axis, we have the keystone taxa, the ones which have the most data points. On the y-axis are the number of studies where a significant difference was found between autoimmune subjects versus healthy controls. The data for autoimmune disease is not as clean-cut as the other conditions I've analyzed over the years for reasons I explained in the first video. However, we still see our clear patterns I continually highlight. For example, we see classic health promoters, which I always mention, like Coprococcus, F. prausnitzii, Lachnospira, and Roseburia, always or nearly always found to be higher in healthy controls. We also see classic opportunistic pathogens, which I always talk about, nearly always shown to be significantly higher in the autoimmune disease subjects. This highlights the dysbiosis I'm always mentioning, which comes up with a large helping of inflammation and gut permeability. So this is at the root cause of autoimmune disease, when also combined with genetic factors and other risks such as viral infections, which, by the way, have been repeatedly shown to cause dysbiosis. For fun, I threw in your two genera of probiotics, Bifidobacterium and Lactobacillus, which have pretty mixed data. I've also included top suspects from the literature, Collinsella, Porphyromonas, and Prevotella, who also have pretty mixed data or very little data, and although may be more of a concern for an individual autoimmune disease, are not determinative across the board. Lastly, we have the genus Megasphaera, which is very consistent, has a reasonable number of data points, is gram-negative, but I can't find any information on this genus and its link to autoimmune disease. Perhaps something to keep an eye on in the future. Similarly, these authors looked at 92 studies in their meta-analysis, and they have summarized the results here in Table 4. They looked at many of the same diseases I did, but have less consistent results. If we just consider genera with 3 or more data points in the same direction, up or down, we're only left with 2. Your probiotic Lactobacillus, which is increased in autoimmune disease, and the genus Streptococcus, also increased, for which I have the most data points, as we take a look again at my chart. When we look at another meta-analysis from different authors, we get these results we see here. On the bottom, circled in red, is our key, which shows us whether a genus was increased or decreased. Blue decreased, red increased, just like the green and orange I use in my chart. As for the genera they found to be decreased in autoimmune subjects, they list many of the great health promoters I talk about all the time, like Ruminococcus, Gemiger, Acinetobacter, Faecalibacterium, Anerostipes, Coprococcus, Allostipes, and Roseburia. What do these all have in common? They are all butyrate-producing, oxygen-sensitive, pH-sensitive members of the two families Ruminococcaceae and Lachnospiraceae. While on the other hand, we see increases in classic opportunistic pathogens such as Enterococcus, Veillonella, Streptococcus, and Negrithella. We also see Lactobacillus here, again, your probiotic acting like an opportunistic pathogen, which so many people are insistent on questioning me on YouTube about my stance on Lactobacillus. Well, keep watching my videos and you'll see this over and over again. Also shown here, mixed in with the bad actors, are Bifidobacterium and A. muciniphila, for which my data is more neutral. But let's be honest, the key is that we're not seeing any of the health promoters I advocate associated with autoimmune disease. They are all on the other side. So now that we looked at the general picture, we'll dive into a few specific autoimmune diseases and highlight some points of note and reinforce some themes, not only from this set of diseases, but themes I highlight in every one of my presentations. Spondyloarthritis is a common chronic inflammatory disease with a disease prevalence of 0.2% to about 1.6%. It mainly affects young people, with around 90% of them developing symptoms before 40 years old. Patients can be classified as axial SPA with predominant involvement in sacroiliac joint and or spine, or peripheral SPA with predominant peripheral manifestation. Ankylosing spondylitis is the prototypic form of axial SPA, and that's what we're going to cover here. Of note, 13% of IBD patients develop AS. When reading the literature, you see the same themes for this, as with the other autoimmune diseases. Nobody knows for sure what the cause is, but is strongly believed to be a blend of genes, viral and/or bacterial triggers, and a ramped-up immune system. Again, we see interleukin-17 believed to be crucial in the pathogenic mechanism of autoimmune diseases. Th17 cells are the main source of interleukin-17, but a number of other immune cells, such as natural killer cells, also produce interleukin-17. As we discussed, this dysregulation doesn't come from out of nowhere. It is an inappropriate response to a pathogen, and this combination attacks the body's tissues. It's just up to luck and genes which determines which tissues get attacked. The ramped up immune response works in conjunction with the other two components, which we covered and will cover in both videos, those of genes and gut permeability. In AS, the HLA-B27, that immune system component of antigen recognition, is the big genetic connection, with 96% of patients with SPA are carriers of HLA-B27, while only 7 to 12% of healthy controls are carriers. That's a huge difference, but let's put that in a different context. Only 5% of the HLA-B27 carriers will develop AS at some point. So that means that to have that genetic predisposition is in no way a guarantee that you will go on to develop AS. It's like Crohn's disease. The NOD2 mutation is the highest genetic risk for Crohn's, and yet it's only present in about 40% of Crohn's patients, and present in up to 20% of healthy controls. And even with identical twins, less than 50% of the time when they have this mutation, they will both have Crohn's. So there is clearly more to this story than just genes. The other component is dysbiosis in the gut, which causes inflammation, gut permeability, and then systemic inflammation with acute or chronic exposure of antigens to the ramped up immune system, which can also lead to our other topic of molecular mimicry, or confusion between friend and foe. To highlight the role of permeability we just talked about, which I reference in about every presentation, we have this paper. This is a biopsy paper, so it is not in my meta-analysis, as I only cite fecal results, but it's still very relevant. We see in Figure 2 a model of the healthy gut, with the cells which line it, the enterocytes, connected firmly to one another to prevent highly antigenic loads from clashing with the immune system. To the right, we see a model of what gut permeability looks like, allowing all sorts of contents to drive inflammation. In chart N, we see significantly more E. coli in AS subjects as compared to healthy controls. Now remember, this is a biopsy paper, so these results are from the cells which line the ileum, not the feces. These are bacteria which have adhered to and invaded the cells, something they mention often in this paper and something I covered in my videos on Crohn's disease and ulcerative colitis. With this pathogenesis, zonulin levels, as seen in G, are much higher in AS subjects, especially AS chronic, as compared to healthy controls. Remember, zonulin is that thing that our bodies secrete in response to a challenge in the gut, which causes the cells which line the gut to separate. And we see that with these two, which show that all of these factors of gut integrity, cadherin, JAM, claudins, and zonular occludins, are significantly reduced in AS. These are all great examples of the connection between dysbiosis, gut permeability, and diseases, in this case, AS, ankylosing spondylitis. This paper too is not a part of my meta-analysis because it does not compare AS subjects to healthy controls. However, I thought I'd include it as it reinforces a couple of classic things. First, we know that in these ulcerative colitis subjects, 19% had axial spondyloarthritis, which if you think about it, that's a lot. And in fact, the more inflammation they had, as measured by CRP, the higher the incidence. Here we see the two poster children for the gut, F. prausnitzii on the side of health and E. coli on the side of disease. As we compare the more severe axial to the non-axial, we see with increasing severity, the microbiome is not the same. E. coli is more abundant in axial, while F. prausnitzii and Eubacterium elegans, another amazing health promoter, are more abundant in the less severe. And if you saw my presentation on stress and the microbiome, we see here, among other things, a significant increase in the ability to steal iron from our cells and give it to the hungry bad guys who love it in order to grow. If you want some proof from my E. elegans statement, here is the data for it across all conditions/diseases I've analyzed. As you can see, it's remarkably consistent. It doesn't have the number of data points as, say, Rosebury or Faecalibacterium for a few reasons. One, it's not as abundant within the microbiome as those two. Two, it's a species and not a genus, which plays into abundance, but even more so plays into 16S technology data limitations. And three, it was reclassified into Lachnospira, so some of its data gets lumped into that genus. Considering those factors, this chart is highly impressive. In this AS paper, we see classic results with a point of note. Here the researchers compared the microbiomes of healthy controls to AS subjects treated with a TNF inhibitor and AS subjects not treated with a TNF inhibitor. This is an important distinction as drugs commonly alter the microbiome for a couple of reasons. So the pre-drug data is more valuable, but the post-drug data is more common for logical reasons. Here we see the classic health promoters, F. prausnitzii, R. bromii, and Eubacterium bioform, significantly higher in the healthy controls versus the untreated AS. We also see classic opposite pathogens, Eggerichella and Clostridium symbiosum, higher in AS, again signifying dysbiosis. But in each case, we see no difference between healthy controls and those treated. The drugs are able to alter inflammation in the gut, and this environmental shift, with its subsequent reduction in oxygen, is able to drive a shift in the microbiome for the better, at least temporarily. Is this addressing the root cause? Well, yes, but mostly no. You have to drive significant shift in the occupants and the environment, and prevent the injury, not take a pill for the pain after you get injured. We also see an outlier, as is usually the case, with Klebsiella pneumoniae, a known bad guy, significantly lower in untreated AS. And lastly, we see a usual autoimmune suspect, P. carpri, also significantly lower in the untreated AS subjects. Now we'll briefly touch on lupus. There's not as many data points for lupus as for some other autoimmune diseases, but it's worth noting nonetheless. Lupus can be difficult to diagnose, and inflammation caused by lupus can affect many different body systems, including your joints, skin, kidneys, blood cells, brain, heart, and lungs. The reason why it can affect so many organs is because it's an autoimmune disease which attacks nuclear components of cells, as in the stuff inside the nucleus, the command center, like the DNA. In fact, serum autoantibodies to your native DNA, that means your DNA, are a specific diagnostic criterion for lupus, and almost every one of your cells has a nucleus. Due to your own cell death during infection, your own nuclear components can be detected by damage-associated molecular patterns, which detect an inappropriate presence of these molecules, and this triggers a response. Additionally, the nucleic acid from bacteria and viruses in infection and in lupus, in particular Epstein-Barr virus, are detected via pathogen-associated molecular patterns, and an inflammatory response ensues, which can contribute to abnormal immune responses through molecular mimicry, epitope spreading, and bystander activation. Therefore, the stage is set by a dysbiotic-driven improper immune response to a trigger, viral or bacterial, resulting in immune cross-reactivity with your own cells. Here we can see in one paper a number of our points, once again illustrated, showing the connection between dysbiosis and disease, in this case lupus. The lupus subjects had dysbiosis as compared to healthy controls, as shown in G and in H, in that their fecal calprotectin, a measure of gut inflammation, and their serum SCD14, a measure of serum LPS, that highly inflammatory component of gram-negative bacteria I'm always talking about, were significantly higher. In addition, our NAVUS levels were 5-fold higher, which is a whole other opportunistic pathogen as it does not contain LPS. In my Crohn's presentation, I talked about how it contains a highly immunostimulatory polysaccharide. Well, here in this study, these authors found that a cell wall lipoglycan was responsible for the antibody response. And as seen here in F, the more R NAVUS, the more antibodies. And in B, the more antibodies to R NAVUS, the more severe the lupus, with significantly more found in those with class 3 and 4 nephritis. Now we'll take a brief look at multiple sclerosis. MS is a chronic demyelinating autoimmune disease of the central nervous system. Approximately 2.8 million people have MS worldwide, and the incidence is also increasing in developing countries and among children. If we're going to talk triggers, then Epstein-Barr virus, think mono, is a proven major risk factor for MS. EBV establishes a lifelong infection in more than 90% of adults worldwide. And, like its sister diseases, herpes simplex 1 and 2, and chickenpox, which can later in life go on to cause shingles in immunocompromised adults, are all herpes viruses. Once you get them, and you'll have at least one of them, more than half of the US are carriers of herpes simplex 1, by the way, then you'll have it for life. You may not be symptomatic, but they are there, hanging out in your nervous system, which is what MS attacks. So this makes sense. You have this ever-present virus, probably causing various levels of chronic inflammation in different individuals. Then in those most reactive, there is a chance for problems. And studies show that patient-derived immune cells, which are autoreactive to myelin, that's the sheathing in MS, that's the sheathing around the nerve in MS, which gets destroyed, are also cross-reactive to a range of Epstein-Barr virus peptides. And yet, only a tiny portion of us go on to be diagnosed with MS, out of billions of us who carry at least one herpes virus. There is certainly a genetic connection as evidenced by studies in family members. However, the main genetic risk factor, once again in HLA, called HLA-DRB1501, is a gene variant carried by 25 to 30% of the population in Northern Europe and the US. But only a tiny fraction of them have MS. And why do twice as many females as males have MS? Clearly there is another factor at play. Sadly, when it comes to the gut microbiome, there aren't a lot of data points in MS, but it appears that the genus Agrothella may play an outsized role in its development. But again, we're talking autoimmune disease. We're looking at the whole picture. I can't change your genes. I have no idea what viruses you'll be exposed to in your life, but I can help you support a proper immune response. I can help you reduce antigenic exposure by addressing your dysbiosis. This is why not everyone with a given HLA or other gene will develop autoimmune disease X, Y, or Z. It's about the whole picture. And the things I can help you with are talked about in just about every scientific paper there is on autoimmune disease. For example, in this paper, I'm just going to start by quoting them. Quote, "Several studies have suggested MS patient gut microbiota has a diminished capacity to produce short-chain fatty acids, i.e. butyrate, which have a known role in limiting the production of pro-inflammatory cytokines and enhancing the production of anti-inflammatory cytokines via an increase in regulatory T cells." We've talked about every component of that sentence. And in figure 7, they have a visual. In here, they show that those without disease progression, or you could argue with those without disease, have an abundance of the two families, Lactobacillus and Oscillosspiraceae. Where have we heard those names before? You've seen this slide in many of my presentations. Those two families are here. Oscillosspiraceae is also Ruminococciae. This is a recent reclassification. These two star-studded families contain the names of health-promoting taxa I mention all of the time, like Ruminococcus, F. prausnitzii, Roseburia, E. rectale, Coprococcus, and Lactobacillus, which contains the E. elegans we highlighted earlier. This slide is accumulation of thousands of hours of work determining the key players in the microbiome. Collected here are the great universal or almost universal health-promoting taxa of the gut. These health-promoting bacteria have been found to be consistently, significantly higher in healthier cohorts and significantly lower in the unhealthy ones across all diseases. The darker the green, the stronger the data as a health promoter. Out of 1,000 or more species in your gut, this handful plays an enormous role in your health. There are a few health-promoting genera outside of these families, like Bifidobacterium, Allostipes, and Odoribacter, which are in other phyla. The goal should be to increase their presence, to control the microbiome. This is my philosophy. This is addressing the root cause. So let's take a look at what a dietary change can do in lupus. Now keep in mind that I pretty much always give dietary advice in my consultations and protocols. But in cases of severe dysbiosis, diet is not enough. We need to drive significant environmental shift in the microbiome by using properly dosed and properly blended prebiotics to suit the needs of the individual. With that said, returning to this paper, these researchers had two groups of clinically similar relapsing, remitting MS patients who had undergone either a high vegetable, low protein diet or a Western diet for at least 12 months. In the high vegetable diet, one of the families I just mentioned, Lactobacillus, was significantly more abundant. That pro-inflammatory interleukin-17 we keep talking about, which is part of the hyperinflammatory response in autoimmune disease, was significantly reduced in the high vegetable group. And the parts of the immune system we've covered, which keep inflammation in check, T regulatory cells and interleukin-10, were significantly increased in the high vegetable group as well. Coincidence? I think not. And there were these tangible results of this microbiome immune corrective action. We see in Table 5 a significant improvement in EDSS, which is a disability scale, and in 3 measures of relapse. I know this is relatively dense material, but I hope this repetition of revisiting the key principles is helping. Just a brief pause here in the presentation. If you could just hit like and subscribe, it would really help this channel out. Now let's touch on type 1 diabetes. The goal here is not to cure your child, although many people are diagnosed later in life, in which case this could be preventative care for some. Once someone has lost their beta cells, that is the insulin-producing cells in the pancreas, they won't be recovered. So again, I'm covering this for preventative purposes, but also to help build an understanding of autoimmune disease, in the event that yours is a different one, which can hopefully be improved. Once again, there is that genetic component here, and once again, we can highlight the HLA. It is estimated that half of type 1 diabetes risk is genetic, and this is even seen in identical twin studies where there is about a 50% concordance rate. In other words, they both have it. But interestingly, this rate rises to 65% by the age of 60. So clearly there is another factor. In fact, the global occurrence rate is growing quickly in those not of European descent. But their genes, they aren't changing. So why do 0.4% of us develop type 1 diabetes? Again, in addition to genes, we once again see a pathogenic trigger. Enterovirus has long been highly associated with type 1 diabetes, potentially causing changes in the microbiome for the worse, like other viruses do, such as COVID. And you'll see in a couple of minutes, dairy proteins are also highly correlated with type 1 diabetes. And this has been known for decades, along with the enterovirus and HLA data. So what we have here is the same story repeating itself. A child acquires an enterovirus, or COVID, or something which alters the microbiome, which results in inflammation, gut permeability, a ramped up immune system, which then recognizes, say, a dairy protein as an invader, a food which is prevalent in the average child's diet. Then cross-reactivity forms due to the molecular mimicry between the dairy protein and the beta cells, resulting in a gradual inability to produce insulin, unknown to the parents until too late. And these authors say what I just said, but a little bit differently in this systemic review, which included 26 papers looking into the association between the gut microbiota and type 1 diabetes. I'm just going to read to you their conclusions word for word, as it's a good summary of what I've been saying. You'll just hear it from the 8 authors in this paper. Quote, in patients with type 1 diabetes, some bacteria increase mucin degradation, results in reduced integrity and increased permeability of intestinal mucosa. That leads to bacterial penetration. The penetration of bacteria into intestinal mucosa leads to stimulation of the immune system and production of antibodies against them. Cross-reaction of these antibodies and surface antigens of pancreatic beta cells, as well as T cell cross-reactivity, results in the destruction of beta cells and formation of type 1 diabetes. And another, butyrate is one of the most important byproducts of microbiota metabolism and plays an important role in colonic regulatory T cell induction, down regulation of pro-inflammatory macrophages, and integrity enhancement of gut barriers through increasing mucin production. And here's another quote. Zonulin is a protein that can be assumed as an important indicator of mucosal integrity and gut permeability. This protein modulates intercellular junctions and macromolecular passage through them. Some bacterial groups can alter mucosal integrity by affecting zonulin. Increase in Bacteroides species and Veillonella species, or decrease in Faecalibacterium species and Roseburia species, correlates with increased serum zonulin levels in type 1 diabetes patients. And here's another. Gut microbiota ingests and ferments fibers and produce short-chain fatty acids. Short-chain fatty acids enter the blood circulation and modulate regulatory T cell differentiation. Thus, autoimmunity is prevented. They mentioned intestinal permeability a lot previously, so let's take a closer look. In type 1 diabetes, the immune system is attacking its own cells, in this case the beta cells of the pancreas. Why? Because of a sequence of events which causes this destruction by friendly fire. And this sequence starts, or in part starts, in the gut. In this study, we see significant differences in gut permeability between the healthy controls and those with preclinical, new onset, and long-term type 1 diabetes. So, what I'm showing you across all of these examples for autoimmune disease, and all of my videos for all of my diseases/conditions, is a consistency of gut permeability, a theme that is constant. Your goal should be to rectify this situation, and that is done by addressing your microbiome. And if not, then things like this can happen. The source for friendly fire due to molecular mimicry can be a virus or bacterium, as we've already well established. But it can also be from food. Food also contains proteins, and with that amino acid sequences, which can cause confusion in the immune system. The #1 most offensive food in this category is dairy. We're not talking about lactose, which is a sugar. We're talking about the proteins. No other protein is introduced earlier in life than that of dairy. So if you're not breastfeeding or doing it exclusively, then odds are you're using a dairy-based formula. You should know that there are numerous studies going back many years associating dairy consumption with type 1 diabetes, like the study we see here. If you look at figure 3, the countries with the highest consumption of dairy products also have the highest rates of type 1 diabetes. In a separate Finnish study, which is the country consuming the most dairy per capita, children who consume the most milk had over 5 times the risk of developing type 1 diabetes versus the lowest consumers. So, while there is a human genetic component to type 1 diabetes, the lifestyle component plays a large role across the board. Not only here, but in studies from other countries like China, which in recent decades has seen an explosion in type 1 diabetes to go along with the explosion in milk consumption. The lifestyle component even appears to be stronger than the genetic one when we look at studies in identical twins, which suggest that only 13 to 33% are pairwise concordant, which means they both have it. So, the factors at play are mode of delivery, the early introduction of immunostimulatory dairy proteins into an immature gut, the quantity and type of dairy consumed, and other factors such as enteric infection. This autoimmune response has to do with dysbiosis, inflammation, intestinal permeability, and genes. And we can see here a couple of the reactive proteins in dairy in another so-called autoimmune disease, ulcerative colitis. Back in the 1940s and 1960s, a number of researchers thought the cause of ulcerative colitis was from dairy proteins. One study claimed that ulcerative colitis was often due to food allergy at least 66% of the time. Another removed dairy foods from the diets of 6 UC patients and all improved. And when they reintroduced dairy, in every instance, it was followed by a frank attack of UC. You can see here in figure 1, also from the 60s, that these researchers found 7 times higher immune reactivity to the dairy proteins casein and lactoglobulin as compared to the healthy controls. Let's take casein for example, which accounts for about 30% of the total protein in cow's milk. And in reality, we're talking more about the A1 beta casein, which studies show is more problematic. There are epitope structures of beta casein which are very similar to epitopes of the glucose transporter 2, which is expressed on our pancreatic beta cells. And autoantibodies to these glucose transporter 2 epitopes have been detected in most patients with recent onset type 1 diabetes. And how do these protein sequences make their way from your gut into general circulation in order to initiate this immunological sequence of events? By gut permeability. And why is there gut permeability? Dysbiosis. Over the years, I've talked with, it seems, too many women who tell me that they have Hashimoto's thyroiditis. From my perspective, it appears epidemic. Hashimoto's is one of the 2 primary autoimmune thyroid diseases, the other being Graves' disease. Although they are both characterized by immune cell infiltration of the thyroid gland, Hashimoto's leads to cell damage and hypothyroidism, while Graves' leads to hyperplasia, leading to hyperthyroidism. We'll just briefly review Hashimoto's. If you suffer from Hashimoto's, you're well aware of the common symptoms such as fatigue, constipation, dry skin, weight gain, dry or loss of hair, and intolerance to cold. Sadly, there is very little data when looking at the microbiome of Hashimoto's, but let's cover what we can. This paper here talks about many of the same principles and notes the guilty connection of various species from Bifidobacterium and Lactobacillus and TPO antibodies. So first, what is TPO? TPO stands for thyroid peroxidase, and it's a key enzyme getting the thyroid to make hormones. And get this, TPO antibodies, as in your immune system attacking your own thyroid enzyme, occur in almost all Hashimoto's patients. You very rarely see an association this concrete. And for those who currently don't have a Hashimoto's diagnosis, but have these TPO antibodies, Their presence may very well raise your future risk. So they argue that data shows that there are human antibodies to protein sequences on a number of these probiotic bacteria, which can cross-react with similar epitopes on TPO via the topic of molecular mimicry we keep highlighting. Now, this is not to say that probiotics cause Hashimoto's. Again, the stage has to be set, because clearly not everyone with these species in their guts, which is most everyone, has Hashimoto's. In fact, one of the accused species here is Bifidobacterium adolescentis, which in my opinion is the most health-promoting species from the genus Bifidobacterium. So, what is setting the stage? If we look at this graphic, we see the same story that every other author and myself are telling you. It boils down to a dysbiotic microbiome, driving permeability, inflammation, and immune dysregulation, where we once again see an imbalance in interleukin-17 and regulatory T cells. They say repetition is key for learning, and so I have more of that here for you. Once again, we'll highlight the connection between gut permeability and a given disease, in this case Hashimoto's. In this study, the researchers compared the plasma zonulin levels of 77 patients to age-matched healthy controls. What did they find? Would you be surprised if I told you that the serum zonulin level, which you now know is a marker for gut permeability, was significantly higher in those with Hashimoto's as compared to healthy controls? Again, this is explained by gut microbial dysbiosis, which not only drives permeability, but also an inappropriate immune response, one that is overreactive, one that has very little tolerance for discriminating between which signals are from a true enemy and which are from self, and thus all can suffer from an unregulated attack. Now let's touch on rheumatoid arthritis, which probably has the most microbiome data amongst the autoimmune conditions in this presentation. In industrialized countries, RA is the most frequent form of inflammatory arthritis. Genetic factors can explain 50 to 60% of the risk, and HLA once again plays a role. However, interestingly, the concordance rate between monozygotic twins is only about 20–30%, which seems contradictory to the 50 or 60% statement from before. If you are among the 1% of us who suffer from RA, I don't have to tell you about the debilitating symptoms of joint swelling, pain, and tendon damage. However, you should know that this persistent, low-intensity inflammatory condition is related to important comorbidities and complications, such as cardiovascular disease, vascular damage, insulin resistance, and amyloidosis. If we look at this diagram, as we already briefly touched on the genetic factors and the end result, bone or tissue destruction, let's now cover what's in between. If you look, you see the same suspects: an immune system which can go two ways, hyperinflammatory or more moderated. If we look to pathogenic triggers, we see a number of suspects. For example, oftentimes in the literature, the species Porphyromonas gingivalis is listed as a prime suspect, which makes sense, as it has been shown to be significantly higher in RA subjects in a couple of studies, and is probably the primary keystone species in periodontal disease, and it is known that periodontal disease is a risk factor for RA. If you follow my videos, I oftentimes talk about oral health and the problems with oral pathogens translocating to other organs where they can be even more problematic than they were in the mouth. Other common suspects are species from the genus Prevotella, but my data shows no consistency here. Other suspects are viral, like several members from the herpes family, as we saw with MS. Like with Crohn's, or UC, or most anything else, there is no one smoking gun. The immune system can react to any pathogen, and the dysregulated hyperinflammatory immune system can cause cellular destruction by friendly fire. In this paper with several inflammatory arthritis conditions, we'll just look at two columns, what's called RA and the one next to it called inflammation. They are the two columns with the most red. Red signifies a positive correlation, blue a negative. As for red, or in other words, the bacteria which are most abundant in RA and inflammation across the board, we see many of the usual bad acting taxa I frequently mention. species from Streptococcus, known bad actors from Clostridium, R. navis, and E. coli. We also see a couple of species from the so-called probiotic Lactobacillus in here as well. Contrarily, for blue, or those taxa inversely associated with RA and inflammation, we see a number of the health promoters I often mention. Names like F. prausnitzii, Roseburia hominis, E. rectale, E. elegans, E. hallei, Anaustypes hadris, and species from Ruminococcus. Again, for like the millionth time in my videos, we're highlighting consistent, statistically significant correlations, which can only mean that each representative taxon is a proven health promoter or opportunistic pathogen. This is an interesting paper for a number of reasons worth pointing out. In any disease, if a person takes a drug, it can alter the microbiome in several ways, which we already touched on. Therefore, these authors analyzed the microbiome and immune markers in those who were recently diagnosed with RA, and then again 3 months later after drug therapy. There are so few papers looking at the microbiome in autoimmune diseases, although for RA there are more than others, and looking at drug-naive subjects has more value as you'll see the changes. Most studies for any condition analyze those on therapy, which is understandable. So here in figure 3, we see how the regulatory immune cells in figure A, the good guys who help prevent autoimmune disease, are significantly lower in the newly diagnosed RA subjects, but are higher after therapy and are even higher in healthy controls. Also in B, you can see the opposite for the pro-inflammatory interleukin-17 we keep talking about, again confirming a pattern proving the connection between a dysregulated immune system and autoimmune disease. Also of note is that I oftentimes find errors in papers. This one has two obvious ones. One for figure 3, the description for A and B is reversed. The other is here. I oftentimes talk about R. navis and how it's an opportunistic pathogen. In my presentation on Crohn's disease, I talk about how this species has been reclassified a couple of times. This paper here is from 2022, but R. navis was reclassified out of Blausia in 2018. So when we read the remarks of the authors like, quote, Blausia navis was the only taxa maintaining significantly higher relative abundance in the ERA patients at endpoints compared to controls, end of quote, we know that it's actually the classic bad guy, R. navis. But the point that they make is a valid one, that the abundance was so high and the bacterium so persistent that it was still very high after the drugs in the RA subjects, while virtually non-existent in the healthy controls. We also see Prevotella, an often accused genus in autoimmune disease, far higher in healthy controls and post-drug therapy, but rock bottom in the newly diagnosed. These are all factors to consider when analyzing the papers. Another often accused microbe in RA is the genus Agrothella, which is pretty much represented by one species, E. lenta. Here we see the data for Agrothella across all diseases/conditions I've analyzed over the years. As you can see, it's almost always significantly higher in the disease cohort and significantly lower in the healthy controls. If we look at the autoimmune column, we see not only is this the tallest, meaning it has the most data points, but it's also 100% consistent, meaning that whenever a significant difference was found in a human fecal microbiome study, it was always significantly higher in the autoimmune. 5 of those autoimmune data points come from RA trials. I'll even supply you with their PMID numbers. For such an abundance of association of data points, you would think there'd be some evidence in humans of molecular mimicry, or some evidence as a trigger for Agrothella in autoimmune disease. But to date, it is exceptionally scant. Even though this is a gram-positive bacterium coming from the same phylum as Bifidobacterium, we know it's an opportunistic pathogen, it can cause bloodstream infections, and, as shown here, has a classic profile of a bad actor. Perhaps its role in autoimmune disease is more of a driving and inflammation approach and gut permeability. The point being that although we don't have all the answers, we do know enough to make a significant difference in the lives of many people. It and the other opportunistic pathogens prefer a certain environment and drive a predictable course of events. We know that the opposite of that looks like, and that should be the goal. And to conclude, we'll end with 3 more papers from the miscellaneous bucket, in case I haven't touched on your particular autoimmune disease of concern. First, we'll start with myasthenia gravis, which is an autoimmune disease which attacks the acetylcholine receptors in muscle tissue, resulting in weakness. These authors cite several studies which show that the production of acetylcholine receptor antibodies is associated with an imbalance in what we've been discussing for 2 videos now, a ramped up immune response versus a dampened immune response mediated by the regulatory T cells. And in figure 2, you can see the significant differences in the microbiomes of the healthy controls versus the MG subjects, with names you should recognize by now. Names like Eubacterium and Faecalibacterium as health promoters, and classic opportunistic pathogens like Streptococcus and E. coli. It is these actors which play an enormous role in determining the immune response, as highlighted in yellow. You need to address them in order to address your autoimmune disease. In this paper, we highlight systemic sclerosis. This data was not included in my meta-analysis as it's mucosal data done during a colonoscopy from 2 sites, the cecum and the sigmoid colon. Interestingly, although they are opposite ends of the colon, the results are very similar. We see an abundance of bad actors like Fusobacterium, Streptococcus, Veillonella, and Klebsiella in the mucus layer of those with systemic sclerosis, while we see a significantly higher abundance of the superhero of the gut, F. prausnitzii, in the mucus layer of the healthy controls. If you see my presentation on opportunistic pathogens or Crohn's disease, you'll understand that this very important mucus layer can be inhabited by a community promoting health or one driving inflammation. Once again, in autoimmune disease, we see a pro-inflammatory profile in their microbiome, which is directly correlated with the disease. And for our very last paper, finally, we have a study which connects a lot of dots. This is a Sjogren's paper. As we can see in figure 5, once again, we see a classic who's who of good versus bad in the microbiomes of the healthy controls versus the Sjogren's subjects. Classic health promoters I mention all the time, like Rhuminococcus, Roseburia, Faecalibacterium, Allostypes, Bifidobacterium, and Lachnospira are significantly higher in the healthy controls, while classic opportunistic pathogens I frequently mention, like Streptococcus, Veillonella, and E. coli, are significantly higher in Sjogren's. Is all of this consistent data a coincidence? Of course not. I have proven this across many hours of videos. You keep hearing the same names over and over again. And I will continue to launch more videos with this very same information. And this consistent imbalance in the Sjogren's microbiome in this paper results in the very consistent finding we've seen thus far. These researchers found that the Sjogren's subjects had significantly higher pro-inflammatory cytokines like interleukin-17 and TNF-alpha. They also found that the Sjogren's subjects had significantly lower levels of the anti-inflammatory cytokine interleukin-10, as well as lower levels of those regulatory T cells which keep the hyperinflammatory response in check. As we aim to address the dysbiosis, we have to appreciate various considerations. For example, if you have a given autoimmune condition, depending on that condition, varying levels of reversal may be possible. If we take type 1 diabetes, for example, I'm sorry, but I won't be able to regenerate those lost beta cells and get you off of insulin. If you have suffered for years from RA and have significant tissue destruction, the amount of reversal, if any, has to be understood. Keys here are understanding the course of these diseases and addressing them preferably sooner than later. Of course, the same is true for many of my videos. For colorectal cancer, it's much better to address your microbiome beforehand when you know you have, for example, say a family history, than later when you have stage 4 cancer. For Parkinson's, obviously it's better to address your constipation, which shows up years before neurological damage. But even with that said, in many studies, to include fecal microbiota transfer studies and the dietary change study in this presentation, many people can experience a reversal in their symptoms to one degree or another. In fact, I have a couple of testimonials that you can view where 2 women I have helped talk about improvements in Hashimoto's. As a person who has a so-called autoimmune condition, Crohn's disease, I have been able to manage it very, very well through my principles before any really nasty things happened, like a fistula or an abscess. So again, better to address things sooner than later. But if you do find this video when things are later on down the road for you, later is still better than never. Not only is there a chance of improvement, but there is also the chance of preventing a further decline, along with addressing any other comorbidities which usually follow, as everything is connected. Any one condition does not happen in a vacuum, something I hope you can appreciate from my videos. So now the ball is in your court. Well, I hope you enjoyed that video, and I will say that basically every single day on my YouTube comments and in emails, I get a lot of thanks from people out there who really appreciate the information I've been sharing. And so part of that is you're welcome. The other part is you can contribute by clicking on the super thanks below. And if you're not doing a consultation with me and you're not purchasing any protocols, it's a great way to support this channel. Each presentation, depending on the presentation, but most of them take an incredible amount of time to put together. There's a lot of material. There's a lot of data checking. And so it's just sometimes 50, 60, 70 hours to put together one presentation. And so if you can just click that super thanks, I'd appreciate that, and we'll keep the information coming.